So in an isometric contraction, there's force generated, but the force doesn't overcome the load, so there's no change in the length of the muscle. So as I push on this wall, I can't overcome the load. I am generating tension in my muscles in my arm, but they're not shortening. So we've got no change in length because the force can't overcome the load. In an isotonic contraction, we can overcome the load. So you generate force that's greater than the load and you move. We've got two types of isotonic contraction that we'll talk about a little later, but you do have a change in length of the muscle. We'll say that our force exceeds the load and the bone moves. Okay, so a motor unit is going to, or understanding what a motor unit is going to help us understand the way that we can create what are called grated responses in muscles, or the ability to change the amount of force based on the task at hand. So a motor unit is defined as one motor neuron and all of the muscle fibers it innervates. So you can have small or large motor units, and what we see in this figure is that we've got two pictured here. This red motor neuron is branching and serving 3 muscle fibers. This purple motor neuron is branching and serving 2 muscle fibers. So they have a different ability to kind of control the specificity of what's going on in the muscle. And if we've got a smaller motor unit, then you have finer control over contractions, as opposed to a larger motor unit where you get greater strength. So in a small motor unit, you have greater control over your contractions. And in a large motor unit, you produce greater strength. Alright, so our motor units, our muscle is served by several motor neurons, and they're all going to produce several motor units, and we will be able to use those motor units or recruit those motor units in a way that will allow us to basically grade the force of our contraction, so we can change it from basically one function or one need to the next. You can change how much force you're producing. So a graded response, or we'll say a graded contraction, occurs as we alter the force in the muscle for varying demands. So to understand graded responses, we have to look at how the different ways that we can test what a muscle is doing. So first we start with a muscle twitch. And this is not something that you experience on a normal moment-to-moment basis. This is something that we can manipulate in the lab to try and understand what's happening with muscles. So what they do is they hook up a frog gastrocnemius to a tension measuring machine, and then you hit it with different levels of stimulus and see how much tension is produced. So a muscle twitch is kind of this lab-created artificial thing that we can kind of understand, or kind of use to understand what is happening in a muscle when it's being stimulated. So a muscle twitch has 3 components, 3 parts to it. The first part is what's called the latent period. And so if you stimulate a muscle and you hit it with some level of millivoltage, there's going to be a little bit of time before you actually see tension produced, and that's the latent period. And what's happening during that time is the events of cross-bridge cycling or excitation-contraction coupling, pardon me. The events of excitation-contraction coupling are taking place, so you don't see any tension built yet. So for our muscle twitch, we can say that the latent period is just after stimulation. We'll have no tension measured. Because the events of excitation-contraction coupling are happening. So it's taking a minute to get that acetylcholine to bind the acetylcholine-gated channels, and for sodium to ultimately cause the end plate potential and begin initiating our action potential. Then, after we have our excitation-contraction coupling events, we will see a period of contraction. During this time, cross-bridge cycling is occurring. Our period of contraction is followed by a period of relaxation where calcium is being actively pumped back into the sarcoplasmic reticulum. So if we look at this on a graph, if we look at our time from stimulus and watch the procession of the events as tension increases, we can see that when you first stimulate the muscle, right at the beginning, nothing happens. There's no tension building, and that's because excitation-contraction coupling is occurring. As soon as that begins, we'll have this increase, this steady rise in contraction, or a steady rise in tension as we go through our period of contraction, and cross-bridge cycling is occurring. And then we have a decline in our tension as calcium is actively being pumped back into the sarcoplasmic reticulum. That's our period of relaxation. So again, this is a muscle twitch, and it's not something that you experience on a moment-to-moment basis. If you've got some kind of neurological problem, you can have a lot of muscle twitches, but this is more just a lab artifact that we use to try and understand the behavior of muscle. So for our graded responses, what we can do is we can change the way that we increase the force in the muscle, and we can do it a couple ways. So if we first start, again, experimentally with this frog gastrocnemius, and we start stimulating it with increasing frequency, we'll see what's called wave summation. And in wave summation, summation always means adding up. In wave summation, we're adding up tension like in a wave. So I'll write a definition for you in a minute. But this is showing us, again, just a muscle twitch, where we have a stimulus applied, then you contract, and you relax, and then you allow the muscle to fully relax, and you don't stimulate it again. Well, if we do stimulate it again before it relaxes, then we'll see that, so this is showing us we stimulate here, we contract, we allow the muscle to start relaxing, but then we stimulate it again. So it doesn't fully relax before it begins to contract again. So that tension is going to build on the last tension. Again, it'll begin to relax, but then we're going to hit it again. And so what's happening is you're increasing tension in this stepwise fashion because you're not allowing the muscle to relax between stimuli. So that's our first type of summation, is what we call wave summation. And what we can say is that the tension in the muscle is going to increase in a wave-like fashion because we're increasing the frequency of stimulus, so there's not enough time to relax before we deliver the next stimulus. So this is going to lead to a phenomenon that we call unfused tetany or tetanus or incomplete tetanus. And so what we're trying to build to when you're really exerting the most amount of tension or a consistent amount of tension across a muscle, we're trying to get to this nice fused smooth tetanic contraction. Leading up to that, we have this unfused incomplete tetanus. So we'll say that this leads to incomplete tetany or unfused. What we can do, another type of wave summation, or wave summation that's increasing in frequency, is going to lead to what we call fused tetanus. So now if we hit the muscle really really fast in very rapid succession. There is absolutely no time to relax. You don't even allow the calcium to start being reuptaken at all. Then what you'll do is you will increase tension, and then there's no time to relax, so there's no calcium is being taken back up, and we'll have this what we call fused tetanic or complete tetanic contraction, and this will last until the muscle fatigues. So, or what this is showing is we're allowing it to relax. We're going to stop here, and so then our calcium will be pumped back into the SR and we'll relax. If you maintain this kind of contraction for a while, or you can keep stimulating it, but eventually it's going to fatigue and you'll have a drop off in tension anyway. So if we say we can also get fused or complete tetanus, and this occurs when you increase the frequency so much that there is absolutely no relaxation period. So in incomplete tetany, you're allowing some relaxation. In complete tetany, you're not allowing any relaxation at all. You're hitting it again and again and again and again and again, and it's got no time to relax between contractions. So we can say that we're increasing the frequency of stimulus so that there is no period of relaxation. The muscle is going to produce maximal tension and fused contraction. So we've got this nice, solid fused contraction. If we do need to alter our level of force so that like I can push this across the table versus like hold 50 pounds in my hand, then we've got another type of summation that's going to happen by changing the strength of our stimulation. So what we just talked about, our wave summation, is occurring because of changes in frequency of stimulation. We can also change the intensity of stimulation, and that's going to give us a different type of graded muscle response. So the next thing that we can do to see how we can grade muscle responses is we can change the strength of stimulation to a muscle. So, and this is going to lead to what we call motor unit recruitment. So to see how different motor units are recruited, we can hit a muscle with different levels of stimulation. The first level that they'll hit it with, like say they hit it with 1 millivolt, and you see no contraction. We call that a subthreshold stimulus. So a subthreshold stimulus is you will stimulate a muscle and observe no muscle contraction. So we're going to ramp up the amount of millivoltage that we hit this muscle with until we hit what's called our threshold stimulus. As soon as we hit our threshold stimulus, we'll see contraction in the muscle, but we're going to allow it to relax also, because right now what we're doing is just trying to see what's varying or increasing our level of stimulus going to do. So at threshold stimulus, this would be the first level of stimulus that you observe contraction in the muscle. Then eventually we're going to be able to increase our level of stimulus. So past threshold, we'll keep up in our stimulus, but eventually we're going to recruit every motor unit in the muscle, and it's not going to be able to produce any more tension. So our maximal stimulus is the level of stimulation that recruits every motor unit in a muscle, so no more tension can be produced. And if we have then recruited every motor unit in a muscle and it's producing its maximal tension, we can keep increasing the strength of stimulation, and we won't see any more tension because it's not possible to see any more tension. So motor unit recruitment is what we're talking about now. So at subthreshold levels of stimulus, you will recruit no motor units. At our threshold stimulus level, we will just recruit the most sensitive motor units. So this is showing, okay, let's say this is 1 millivolt to that motor, or from that motor neuron. That is not enough to produce tension, so no motor units are going to be recruited. Okay, so we hit it with 2 millivolts, still not enough to stimulate a motor unit, so we are not producing any tension. When we hit it with 3 millivolts, now we're going to stimulate our first motor unit, so it must be the most sensitive and will produce a little bit of tension in that muscle. So our threshold stimulus recruits most sensitive motor units. Okay, now I hit it with 4 millivolts, and I'm going to recruit another motor unit, so I'll get a little bit more tension. Hit it with 5 millivolts, recruit another motor unit, build a little more tension. Hit it with 6 millivolts, recruit more motor units, build a little more tension. Now, as soon as I hit it with 7 millivolts, I've recruited every motor unit in this muscle, so it's going to produce the maximum level of tension that it can. Now we let it relax, I hit it with 8 millivolts. Well, I don't have any more motor units to recruit, so it's going to produce the same maximal level of tension. So our maximal stimulus is going to be the level that is going to recruit all of our motor units in the muscle, so they're the last ones to be recruited are the least sensitive. So this is going to be another way that you can vary the amount of strength depending on your task. So if I'm just writing and using the muscles in my fingers to write, then I am using just my most sensitive motor units. If I'm trying to curl that 50 pounds, then I'm recruiting every motor unit in my muscles, and they're producing maximal contraction so that they can bear that load. So again, our graded responses are going to give us varying amounts of control over what we're doing with a muscle. Okay, so we briefly talked about isometric versus isotonic contractions at the beginning of class. Isotonic contractions, again, are those contractions that are changing the length of the muscle because the force that we're applying is overcoming the load. We've got two types of isotonic contractions. The first are called concentric, and in concentric contractions, your muscle is shortening. So in isotonic, we see a change in the length of the muscle. And in a concentric contraction, we see shortening. So if I'm looking at my biceps brachii, and the contraction is bringing my cup to my mouth, then this is a concentric contraction. And when my muscle is shortening as I bring that cup to my mouth. Now, as I set that cup back down, my muscle is lengthening. That's eccentric. So first, concentric, you can see what happens. We are going to give this, this is the frog gastrocnemius I was talking about. We're going to give it some load to bear, and it's going to shorten. In an eccentric contraction, our muscle is going to lengthen. So, that's not an eccentric contraction. In an eccentric contraction, that would be like putting your cup back down. Now we're generating tension, but our muscle fiber is lengthening. That's eccentric. So for concentric, we can say like biceps. The biceps brachii are undergoing a concentric contraction when you're lifting your cup to your mouth. And they are undergoing an eccentric contraction when you're setting the cups back down. So it's an eccentric contraction. If you want a little tip to help you remember which is which, if you're an eccentric person, you're really big and out there. And you're lengthening your muscle in an eccentric contraction. That may or may not help you. In an isometric contraction, we're applying some force. Your muscle is contracting, but we're not changing in length. And that's because our load is overcoming the muscle's tension-producing ability, basically. So the muscle is attached to a weight that exceeds the muscle's peak tension-developing capacities, so the tension increases, but the muscle doesn't shorten. All right, so what are different things that can influence muscle contraction? Well, the number of muscle fibers stimulated. So how many, as you increase more of the muscle fibers that are being stimulated, you'll increase the amount of tension in the muscle. The size of the fibers is also going to have an effect. So if you stimulate a bigger muscle fiber, it's going to be able to produce more tension. The next thing is frequency of stimulation. If you stimulate it and don't allow it to relax in between, then we'll have that summation of our tension and produce more tension. And then also the degree of muscle stretch, which is kind of weird to think about. We'll look at a figure in a second that shows it. So this figure in your book sums all of those things up. If you increase the amount of muscle fibers recruited, you increase your contractile force. If you increase in size of the muscle fibers that you're bringing on, so if we stimulate a larger muscle fiber, we will increase our contractile force. If we increase the frequency of stimulation to a muscle, we'll increase our contractile force. And then if we increase or stretch our muscle to this ideal level, we'll be able to increase our contractile force. So there's this ideal length-tension relationship in our sarcomeres that's going to allow a muscle to produce the highest levels of force. So if you look at it if we've got lots of overlapping, so our sarcomeres are shortened, then we've already got some overlapping of our thick and thin filaments. We're not going to be able to produce lots of tension because there are already some overlapping, so it's not going to help. If we've got this at our resting length, it's kind of one of our optimal length where we've got this optimal overlapping of thick and thin filaments. Now our thick filaments are going to be able to pull on those thin filaments and generate the maximal amount of tension. As we stretch it a lot, so there's this kind of length where you can stretch it some and that will increase our tension, but if you stretch it a lot to where there's almost no overlap of thick and thin filaments, then it's going to be really hard to produce tension, because if your thin filaments are very far away from your thick filaments, they can't grab onto it. So just be aware that there is an ideal length-tension relationship.